Polysaccharides are molecules that are made up of 3 or more monosaccharides that are connected by glycosidic bonds. We're going to look at 3 different types of polysaccharides, starting with cellulose. Cellulose contains hundreds to thousands of glucose molecules that are connected by glycosidic bonds, which we're going to look at in a second. And you can see for cellulose that the way that the glucose monosaccharides are connected, it creates like a string or a strand of glucose molecules, a nice linear connection of them. Humans can't digest cellulose, which means that we don't have the enzymes necessary to break the glycosidic bonds in cellulose. So cellulose's main job is to make up the cell wall and also provide structural support for plants. Let's look at the glycosidic bond. So cellulose contains beta 1 to 4 linkages, or 1,4 beta linkages. And now you know what that means, you'll be able to look at this structure and understand. So this is the glycosidic bond. The connection is between this carbon atom, which is carbon number 1 of its ring, and the other connection is right here, which is carbon number 4 of its ring. So that's the 1 to 4 notation. I wrote 4 2 times. And then in terms of the beta notation in this, we're looking at carbon number 1, and we're also looking at carbon number 4, and we're asking ourselves, are either of those, carbon number 1 or carbon number 4, are either of them anomeric? Carbon number 1 is anomeric. Remember, anomeric just means that it is the very right-hand point of its own individual ring. So here's the anomeric carbon of our other ring. The anomeric carbon is carbon number 1, and carbon number 1 has its oxygen in the up position, so it's the beta anomer, which is where we get that beta 1 to 4 notation for the glycosidic bond. So next up, we're going to talk about starch. Starch is found in corn and wheat and potatoes, so it's also a plant polysaccharide, but it is digestible. So we are capable of breaking the glycosidic bonds that make up starch molecules. There are 2 different types of starch polysaccharides. One is called amylose, and the other is called amylopectin. And they are different only in terms of their glycosidic bonds. Both amylose and amylopectin are made up exclusively of glucoses, so it's just a bunch of glucose monosaccharides. And it is the way that plants store glucose within their structures. This is digestible, as I think I said that already, so it's digestible by humans. For amylose contains only 1 to 4 alpha glycosidic bonds. So here would be a close-up of the structure of amylose, and we can see that the 1 to 4 notation comes from the numbering of the 2 carbon atoms that are connected by the glycosidic bond, carbon number 1 of one ring and carbon number 4 of the other. The alpha notation, we focus on carbon number 1 and carbon number 4. We ask ourselves, is either of those carbons anomeric? Meaning, are either of those carbons the right-hand point of the ring? Carbon number 1 is anomeric, and it is the alpha anomer because its bond to oxygen goes down. So that's how we get the 1 to 4 alpha notation. This, if you look at this over here, so this might be notation that you have not seen before. So we've got this square bracket, and then outside of the square bracket, it says 300 to 600. So what that means is the monosaccharide that's inside that square bracket is repeated anywhere from 300 to 600 times to make up an amylose molecule. Now, the other type of starch is amylopectin. And although it also contains glucose, the difference between amylopectin and amylose is that amylose contains, in addition to the alpha 1,4 glycosidic bonds, it also contains some alpha 1,6 glycosidic bonds. So let's number these two monosaccharides that are connected together. There's carbon number 6, and there's carbon number 1. And in terms of designating alpha, carbon number 1 is the only one that's anomeric here, and it is the alpha anomer. So because amylopectin has this type of linkage right here, which causes the glucose molecules to arrange linearly, same as amylose, it also has this type of linkage right here, which causes basically some branching in the molecule. So it gives it a more interesting structure. It's not just purely a straight chain. And the last type of polysaccharide that we're going to talk about is glycogen. Glycogen is the way that animals, including humans, store glucose. So glycogen is also made up exclusively of glucose monosaccharides. And it is, as we talked about this before, it is how we store excess glucose that we take in from our diet. So glucose that we don't need for energy, we store as glycogen. And we've talked about how there's limited glycogen storage inside a human. So if we reach that storage limit, then we start storing glucose in our fat cells. Now, glycogen is pretty similar to amylopectin, but it is extensively branched. So it's the same kind of idea. Let's look at its glycosidic bonds. It has 1,4 alpha glycosidic bonds, and it also has the 1,6 glycosidic bonds. But the main difference between the amylopectin and glycogen is that we just see more instances of the 1,6 connection. So we see just a greater amount of branching. It just happens more frequently than what we see in amylopectin.